You know, back in 2018, the Nobel Prize in Medicine celebrated this just massive win.
Oh, absolutely. The immunotherapies that reactivate the immune system.
Right. Finding ways to get the body to hunt down cancer. It really felt like we had finally cracked the code on how tumors evade our defenses.
It did feel that way for a while, yeah.
But what if we've been completely blind to a second, far more insidious stealth mechanism?
That is exactly what we're looking into today.
Because today, we are taking a deep dive into how cancer cells are physically burying themselves under this, microscopic, sugary invisibility cloak.
Yeah, it sounds like science fiction, but it's very real. We're unpacking a groundbreaking 2026 study from Cell Genomics by Daly and colleagues.
And their mission was mapping the genetic switches behind something called cancer glycome remodeling.
Right, and that term glycome remodeling, I mean, that is really the frontier of oncology right now.
So what exactly is the glycome?
Well, the glycome is this dense, incredibly complex forest of carbohydrate structures, and it coats the surface of literally every single cell in your body.
Just a forest of sugars on the outside of the cell.
Exactly, and for a long time, we thought of these sugars mostly as structural, maybe just cellular scaffolding.
But this new paper proves something completely different, right?
Yeah, it proves that cancer cells are actively weaponizing this forest to survive. They are radically rewiring their internal factories to produce specific sugars.
Sugars that basically trick the immune system into ignoring them.
You hit the nail on the head.
Okay, so to give you listening a visual, think of a cancer cell like a rogue bakery.
A rogue bakery. I like that analogy.
Right, so all the dangerous mutated proteins, basically the evidence of its rogue status that's all operating inside.
Boy, hiding out of sight.
But to keep the health inspectors out, and those are your patrolling immune cells, the bakery coats its entire exterior in a very specific thick layer of sugary frosting.
And in human biology, that specialized sugar frosting is made of molecules called sialic acids.
Sialic acids, okay.
Yeah, and to the immune system, a thick layer of that sialic acid acts as this giant flashing do not eat me sign.
That's wild. And those health inspectors, the immune cells, how do they actually read the sign?
Well, they use a family of immune receptors known as Siglecs.
Siglecs.
Right. Stands for sialic acid-binding immunoglobulin-like lectins. These receptors are mounted on the outside of your immune cells, just constantly sweeping the environment.
So when they bump into a cell, they're literally tasting the surface chemistry.
Exactly. And if they taste that dense sialic acid frosting, it triggers this profound cascade of suppression inside the immune cell.
I always keep a mnemonic handy for this interaction, just to lock it in for you listening. It's Siglec senses sialic acid to suppress.
That's actually a perfect way to remember it.
But how exactly does sensing a sugar on the outside paralyze a cell on the inside? I mean, there has to be a physical mechanism there.
Oh, there is. And it's just a brilliant piece of evolutionary engineering. When the Siglec receptor binds to the sialic acid outside, the mechanical force causes the receptor's tail, which hangs down inside the immune cell, to change shape.
Wow, so it physically twists.
Yeah, and this structural shift exposes a sequence of amino acids called an item motif.
An item motif?
Right, an immunoreceptor tyrosine-based inhibitory motif.
So it's essentially like a biological panic button being exposed.
Exactly. Once that item panic button is exposed, a chemical tag is added to it. We call that being phosphorylated.
Okay, and what does that tag do?
It immediately calls in an enzyme named SHP1. And SHP1 is a powerful molecular brake.
It stops everything.
It physically sweeps through the interior of the immune cell and dismantles the signaling pathways required for an attack. The cell might be fully armed, but that SHP1 brake renders it totally inert.
Wow. Okay, so if we step back for a second, this completely flips the script. The cancer is actively manufacturing this armor to hit the off switch.
It's proactive defense, yeah.
So here's a quick summary for you listening. Cancers coat themselves in sialic acid to hit the off switch on immune cells via these Siglec receptors.
Perfectly summarized.
Which brings up a question for you all. Have you ever thought of a simple sugar molecule as a weapon of stealth? It totally forces us to rethink biology.
It really does.
But here is the thing that really bothers me about this timeline. If these sugars are so critical, why did it take until 2026 to find the genes responsible for building them?
Well, the bottleneck was entirely technological. We simply didn't have the right screening tools.
Really? Because we've had CRISPR for a while now.
We have, but up until very recently, the gold standard was a loss of function, or CRISPR screen.
Oh, right, where traditional CRISPR acts like molecular scissors.
Exactly. The logic seems sound right. You go into a cell, cut a specific gene to turn it off, and then see if the cell stops making the sugar armor.
Right. If the armor disappears, you've found your culprit.
But there are two massive limitations. First, if you cut a gene that is essential for the cell's basic survival, the cell just dies instantly.
Oh, right. So the whole factory burns down and you learn absolutely nothing about the sugar production.
Precisely. And the second limitation is even more restrictive. Cell surface glycosylation, the way a cell builds its sugar forest, is highly specific to the tissue.
So a lung cell builds a different forest than a liver cell.
Exactly. Because of this, roughly 50% of all the genes capable of building these complex sugars are completely dormant in any given healthy cell.
Wait, half of them are just turned off?
Yeah, and you cannot learn what a switch does by breaking it if that switch is already in the off position.
Right. I mean, breaking a light switch that is already flipped off doesn't tell you what room it illuminates.
Exactly.
Okay, let's unpack this. Instead of turning the lights off, what if we wire every single switch to stay permanently on?
And that right there is the breakthrough at the heart of this paper. They bypass the scissors entirely and utilize CRISPR activation, or CRISPRa.
CRISPRa. Okay, how does that work?
They engineered a specific leukemia cell line, K562 cells, and they introduced a dead Cas9 protein, dCas9.
A dead Cas9.
Yeah, it retains its incredible ability to navigate the genome and park next to specific DNA targets, but its cutting mechanism is permanently disabled.
But just parking next to a gene doesn't turn it on. How are they forcing the overexpression?
They fused this dead Cas9 to a powerful transcriptional activator called a VP64 domain. VP64 acts as a massive molecular megaphone.
Oh, wow.
Yeah, so when the dCas9 parks next to a target gene, the megaphone aggressively forces the cell's machinery to read and translate that gene at maximum capacity.
That is brilliant. So how many genes did they target?
They unleashed a library containing over 104,000 different guide RNAs into millions of these leukemia cells.
104,000. So it's just this massive, chaotic soup where every single cell is forcefully overexpressing one random gene.
Exactly.
But once you have that soup, how do you isolate the ones that successfully built the invisibility cloak?
They bathed the entire population in fluorescent probes designed to perfectly mimic human Siglecs 7, 9, and 10.
Oh, so the ones with the right sugars will glow.
Right. Then they ran the cells through a fluorescence-activated cell sorting, or FACS, machine. It separated the cells that glowed the brightest from the ones that glowed the least.
And by sequencing the DNA of those brightest cells, they could trace back which forced gene caused the massive sugar buildup.
Exactly. And they identified 941 distinct genes that modulated this binding.
941. That is a huge hit list.
And the truly crazy part, over 50% of those were normally totally silent in these cells. Traditional CRISPR would have missed them completely.
That is insane. So, reaching our 5-minute milestone here, let's do a quick recap and bullet summary. Bullet 1. Cancers use sialic acid sugars to trigger immune-suppressing Siglec receptors.
Yep.
And bullet 2. CRISPR screening, which overexpresses genes, reveals hidden regulators of this process that traditional knockout screens completely miss.
That's the perfect summary.
So, question for you listening. If you were hunting for a hidden defense system, wouldn't you want a tool that forces every possible weapon out of hiding?
You'd be crazy not to.
But, finding the 900 parts of the factory doesn't tell us how the assembly line actually operates.
No, it doesn't. But when they mapped out the functions of these genes, they uncovered a massive tug-of-war happening. We call it biosynthetic competition.
Biosynthetic competition. Wait, so the genes are actively competing for the same molecular foundations?
They are. Building a complex carbohydrate on a cell surface is a multi-step, highly contested assembly line.
Okay, walk me through the assembly line.
Well, you have a base structure, usually galactose. On one side of the tug-of-war, you have the ST3GAL family of genes.
ST3GAL, what do they do?
They encode enzymes called sialoglycans. Their sole imperative is to grab a sialic acid molecule and slap it onto that galactose base. It's like putting a final chemical cap on the sugar branch.
Okay, so one worker, ST3GAL, wants to put a cap on the product and ship it out as a Siglec ligand. Who are they competing against?
They're competing against the GCNT and B3GNT families. These enzymes want to elongate the sugar branch by adding a molecule called GLCNAc.
So they want to keep building long, complex branches, while ST3GAL just wants to cap it and be done.
Precisely.
Yeah.
And whichever gene is louder, whichever one is overexpressed, wins the tug-of-war.
It's literally a race for the same foundation.
Yeah. And the screen proved that different Siglec receptors demand completely different architectural styles from this assembly line.
Right, like Siglec 7 preferred what we call O-linked glycans, which are driven by ST3GAL1 and 2.
Exactly.
But Siglec 10 had a totally different preference, right? It preferred N-linked glycans.
Yes, driven by ST3GAL4, 6, and a novel hit, ST6GAL1, which attaches the cap at a slightly different geometric angle.
You know what's fascinating to me? What was totally missing from the hit list?
Oh, you mean the fucosylation genes?
Yeah. Entirely missing were genes for adding fucose, or synthesizing the raw CMP sialic acid. It proves those are totally dispensable for this immune evasion.
It just highlights the dark efficiency of cancer. It doesn't waste energy reinventing the supply chain. It just hijacks the final competition between capping and branching.
So just summarizing this section, the amount of immune-suppressing sugar on a cancer cell depends on a fierce competition between capping enzymes and branching enzymes.
That's the core of it, yeah.
So knowing this tug-of-war exists, I want to ask you listening, which side of the rope do you think your own healthy cells are pulling on right now?
Well, in a healthy state, it's an exquisite balance. Cancer just floods the zone to win the game.
Right, but even if the assembly line builds the perfect sugar, it can't just float in space. It needs scaffolding, right?
Exactly. In glycoimmunology, we call these scaffolds professional ligands. There are specific surface proteins that present the sugars at just the right density.
Like little display racks.
Exactly like display racks. For Siglec 7, the hits were mucins, specifically CD44 and MUC4.
And MUC4 is infamous in pancreatic cancer, right?
It is. It's a highly aggressive driver.
Yeah.
And this gives us a direct mechanical link to why it suppresses the immune system so well.
That is wild. And what about Siglec 9?
For Siglec 9, the hits were CD44 and CSPG4, which often carry a rare modification called chondroitin sulfate.
Okay, here is where it gets really interesting. We have to talk about busting the CD44 myth.
Oh, this is arguably the most disruptive finding in the whole study.
Because past studies universally claim that Siglec 10 binds exclusively to a protein called CD24. Like that was textbook doctrine.
It was. But in this completely unbiased genome-wide screen, CD24 wasn't a hit at all.
0 hits.
0. They even forced the cells to overexpress CD24 just to double-check, and it did absolutely nothing. The leukemia cells didn't even have CD24 to begin with, yet they bound Siglec 10 perfectly.
Okay, so if the scaffold isn't CD24, how did they prove what Siglec 10 actually cares about?
They proved it cares broadly about N-linked sugars. They took an acute myeloid leukemia model, or AML, and knocked out the ST3GL4 gene.
The one that caps N-linked sugars?
Exactly. And Siglec 10 binding evaporated. Then they knocked out ST6GAL1 in a multiple myeloma model, doing the exact same thing.
Wow. Absolute proof that the sugar, not the CD24 scaffold, is the true key.
It's a profound lesson. The protein scaffold is basically dispensable.
Okay, 10-minute milestone. Let's do our second bullet recap. Bullet 1. Assembly line competition dictates sugar production on the cancer cell surface.
Correct.
Bullet 2. Professional ligand proteins, like mucins, act as display racks for these sugars. And bullet 3. Contrary to popular belief, CD24 is totally dispensable for Siglec 10 immune suppression.
A huge paradigm shift.
So, question for the audience. How often do we blindly accept a scientific fact like the CD24 connection, only to have a new data set completely rewrite the textbook?
We always have to be ready for new data to destroy old models.
So true. But how does this translate to saving lives? I mean, what's the clinical side of this?
Well, they cross-referenced their CRISPR hits with the Cancer Genome Atlas, the TCGA clinical survival data to find the real-world villains.
And they found a terrifying correlation in low-grade glioma, or LGG.
Yes. Patients with high mRNA expression, high copy numbers, or low methylation of a gene called GAL3ST4 showed dramatically accelerated disease progression.
And really poor survival rates.
Extremely poor.
So what does GAL3ST4 actually do in brain cancer?
It encodes a sulfotransferase. It adds a highly negatively charged sulfate group directly to the galactose. They knocked it out in LN229 brain cancer cells, and Siglec 7 binding plummeted to just 20%.
20%. Okay, let's use a visual analogy. Imagine the sugar structure as a specific lock.
Okay, a lock.
In most cancer, sialic acid is the only key needed. But in brain cancer, GAL3ST4 adds a second keyhole, a sulfate group creating a novel 3-sulfosixylochlore 1 structure.
That's a great way to describe it. It's like two-factor authentication.
Yes, two-factor authentication. Without that sulfate, the brain cancer cell can't turn off the immune system.
It's an incredible discovery of tissue-specific immune evasion.
15-minute milestone. Let's recap. Bullet 1. LGG patient survival is tightly linked to the GAL3ST4 gene. Bullet 2. Siglec 7 requires this unique sulfation mechanism to bind and suppress the immune response in brain cancer cells.
Spot on.
So audience, could designing a drug to block this one specific sulfate enzyme be the master key to treating deadly brain tumors?
If we connect this to the bigger picture, that is exactly where we're going. We are moving toward a future of precision glycoimmunology.
Moving away from one-size-fits-all treatments.
Exactly. If a doctor knows a patient's exact tumor expression profile, whether it's ST3-GAL4 in leukemia or GL3-ST4 in glioma, they can predict exactly which immune checkpoints to block.
That is just incredible. But before we go, I want to connect directly with you listening with this mind-bending detail buried in the paper's limitations.
The mouse models.
Yes. The researchers point out that Siglec receptors are not conserved in mice.
No, they're not. And murine immune glycosylation is drastically different from humans.
It's a totally different sugar code, yet we rely on mice to test all our cancer drugs.
We do.
So I want to leave you with a final thought to mull over. If mice have a totally different biological operating system when it comes to these sugars, how many miraculous cancer cures have we missed simply because we were testing them on the wrong hardware?
It really makes you wonder.
It really does. Thank you all so much for taking this deep dive with us today. Keep questioning the consensus, and we'll catch you next time.